changer is slide changer. What about the remaining, or if you are there? Good afternoon. So it's a post-lunch session. Everyone is getting sleeping, and already maybe 30-40% absent. Okay. So today we'll give some demonstration on some of the instruments we have in our laboratory. Instrumentation lab is the HPLC, another one is the FTIR. So before entering into the demonstration or handling the instrument, just have a look at the HPLC, the instrumentation part. Okay, and what is the major applications? Why we need to use these chromatography techniques is very familiar in pharma industry now. Okay, so the pharma industry is a highly regulated industry. Okay, and each and every component we are using for the manufacturing of any dosage form, okay, should be characterized qualitatively and quantitatively every step, not only during the manufacturing process till their expiry date. Every moment we need to check the qualitative and the quantitative characterization of all the components which are involved in the manufacturing process, not only the API. For example, you are manufacturing paracetamol tablet. You are not only characterizing the only paracetamol, the major constituent API, but also all the diluents, what kind of binders we are used, what kind of sweeteners we are used, what kind of disintegration we are using, okay, what kind of stabilizers we are using. And also there is a case of the process impurities and the stability impurities. The process impurities will arise during the manufacturing process. Stability impurities will arise during the storage time. Okay, so all these constituents we need to analyze qualitatively and quantitatively. Okay, so if you look at the marketed products in entire the globe, more than 95% of the molecules are enzyme inhibitors or enzyme stimulators. Okay, if you look at our human body is just like a biochemical reactor. Each chemical reactor requires one enzyme. So sometimes the enzyme may be overactive and sometimes maybe enzyme may be low active. In each condition that leads to disease. 90% of the drugs we are using in the market are either enzyme stimulators or enzyme inhibitors. So these enzymes, if you look at enzymes that contains the proteins, amino acids, the chemical structure of the enzymes, to inhibit the enzyme or to stimulate the enzyme, we require the organic molecules, the molecules that contains organic in nature. So that the molecule that contains the binding affinity towards the enzyme we are taking as a drug target as a receptor. The receptor that contains a combination of some amino acids. For example, if you look at some of the receptors of angioconverting enzyme that contains a molecular weight of 8,000, and that there is a series of 8,000 means, for example, some 1700 to 1600 amino acids are in the sequence, just like a chain. Okay, the amino acid of 14th amino acid, 55th amino acid, and 620th amino acids pull just like a ribbon-like structure, and it forms the active site. So these active site contains the amino acids in the sequence of 51, 151 and 1700 like that. So it combined these amino acids becomes the active site. So our drug molecules enter into the systemic circulation, distribute the entire body and reach it to the receptor where it is allocated, the receptor, and interact with that active site. Very competitively. There is there's different kinds of binding affinities. So after binding that particular molecule to the enzyme or the receptor, it will inhibit or that will stimulate. Some of the drugs we require angioconverting enzyme inhibitors, means the molecule captopril or enalapril will go and bind with angiotensin converting enzyme and they will inhibit the activities of the enzyme. That will get some biological effects. Some of the enzymes will stimulate the activity. Okay, a thyroid hormone, there is a stimulating the activity. Some of the molecules are binding and stimulating the activity. So ultimately, most of the times, these all organic molecules are behaving as a enzyme stimulators or enzyme inhibitors. Most of them are the organic molecules. Okay, so these organic molecules we need to characterize every stage. During the way, for example, organic molecules, very first we need to synthesize or we need to extract from the natural source. Okay, these are all organic in nature, but we don't know the structure, exact structure when you're dealing with the new molecule. For example, we synthesize paracetamol. Paracetamol has been synthesized long back, maybe say 50, 60 years back. Okay, very fast. When the paracetamol is synthesized very fast, they need to understand the structure, or they need to confirm the or elucidate the structure by means of a different spectroscopic methods, a combination of different spectroscopic methods. When we evaluate the structure of any molecule, that is a new molecule, by NMR or by X-ray crystallography structure, by mass spectrometry or by FTIR or by the combination of all the spectroscopic methods we'll utilize to characterize very fast. Okay, so we establish the structure of the molecule. Though this molecule that contains a benzene, there is a third position, there is a chain, this is a fourth position, there is a chain. We need to characterize the molecule. Not only the organic molecules, what are the enzymes or the receptors you're studying in the pharmacology? Okay, how can you say that this enzyme structure is this one? Okay, this is active site. Okay, how can you say that any mechanistic properties of any drug? You're studying mechanistic studies. How can you say that the mechanism of the captopril is by inhibiting the angiotensin converting enzyme? First, we should know the structure of angiotensin converting enzyme structure. Okay, we need to isolate from the biological system. We need to characterize by means of NMR and mass and combination of all these studies. We have to characterize. This is molecular formula. This is the engine ring. This contains this functional group. This contains this function. We have to characterize, fully characterize these organic molecules. Okay, once it is characterized, then we can only think is we don't require to every time we need to characterize qualitatively, only the quantitatively. Okay, by using the existing methods, we can do quantitative analysis of these molecules. Okay, if the molecule is a pure, directly we can go for the UV spectroscopy for assay, or directly we can go for the fluorometry, or directly we can go for the mass spectrometry or anything for the quantitative analysis. But the problem is in pharmaceutical industry, all these molecules are coming with different constituents that contains paracetamol is combining with different impurities and combining with different diluents which we are usually using in the formulation process. Okay, so we can't go directly UV. For example, you would be taking the tablet and dissolving for a suitable solvent. If you keep in the UV spectroscopy, we can't. That contains 5 to 6 constants. Each constant will get the absorbance. We can't say that the absorbance is because of paracetamol. Even that contains a paracetamol is 90% of the constituent is paracetamol, but the absorbance is because of that contains a sugar, that contains a binding agents, that contains any other disintegrating agents, that contains impurities. The absorbance is maybe because of different constituents. Okay, so in that case, when you are handling with the any sample that contains more than one constituents, wherever we require to go for the chromatographic analysis before entering into the analysis. As the chromatographic analysis means the chromatography, as you know, that is a separation technique. First, we need to separate all the constituents. Then you have to submit the constituent one by one to the detector. Okay, the detector follows again the spectroscopic methods. There are two types of analytical methods. One is a spectroscopic method. Another one is a chromatographic methods. Spectroscopic methods means like UV, IR, wherever the light we are using. Spectroscopy, light. We are using the light to determine qualitatively and quantitatively based upon some of each instrument will follow each technique will follow a different defined mechanism of action and theory. Okay. So, for example, we look at the chromatography techniques. Again, we have HPLC, we have GC, and we have hyphenated techniques like LCMS and GCMS. We have paper chromatography technique, we have column chromatography technique, we have TLC. We have different chromatography techniques. Okay, so here I think you have understood why we need to go for the chromatography. Okay, whenever you are handling the component that contains a mixture more than one component, directly we need to go. First, we have to separate it, then identification. The chromatography involves the separation and identification. The UV spectroscopy involves only identification by qualitative or the quantitative. Spectroscopy involves the first part is separation. And detector, HPLC contains UV again. HPLC, the instrument, if you look at, but one of the part is UV again. We are separating the first part. and one by one the molecule is make it available to the detector UV spectroscopy. You will get the absorbance for the UV absorbance. Okay, so that's why the spectroscopic method, the chromatographic methods have a vast applications in pharma industry, because in the pharma industry we are dealing with the constituent that contains the multiple constituents. If you take any sample that contains the multiple constituents, even the pure sample of API also we have to run the API with the spectroscopic chromatographic method, because that API that contains not only the paracetamol, that also contains the stability impurities and the process impurities, and also some other impurities which are arise during the process of manufacturing process. Not only during the manufacturing, but also till its expiry date. So, for example, Dr. Reddy's is manufacturing 50 lakhs tablets of paracetamol in batch number 1 to be marketed in the Indian market. Today, for example, the 22nd of March 24, they are manufacturing 50 lakhs tablets. The expiry date is 2027 March. There are 3 years of time. Means the 50, they never send all 50 lakhs tablets into the market immediately. They will take some samples from the batch number. On the batch number, they will collect some 100 to 200 molecule to 200 tablets and keep in the store. The store is, for example, these 50 lakhs tablets are going to the Indian market. In the store, they will create the Indian climate conditions. What is the usual Indian climate conditions? It is maximum temperature of 45 and minimum temperature of 5 degrees or 4 degrees. They will maintain these temperatures for the next 3 years. Okay, so every 6 months they will collect that paracetamol tablet and they will test for the qualitative and quantitative analysis. The day one, how we got the results? After 6 months also we should get the same result. After 1 year also should get the same result. Till entire expiry date, they have to collect the samples every 6 months and they have to run the test. If something is happening, there is a company's responsibility that all the remaining unused tablets should bring to the again company, and they have to destroy that. So this one here, this is called as a regulatory process, not only during the manufacturing process, but also the entire time. That it is applicable for all the formulations, not only the tablets or capsules, all formulations or injections or anything, even any kind of formulation. Okay. So here we need to, for the analytical purpose, each regulatory body will specify the test. US FDA, you have to determine qualitatively the paracetamol by USP of the method of so-and-so HPLC method only. Okay, so for example, Dr. Reddy's is doing the HPLC method of paracetamol. That will be recorded in the dedicated server of instrument waters. We can't do any manipulation. The US FDA people will not see our data. They will see the dedicated server from the app, that is the waters company. Today I am performing an experiment. I am weighing this one. I got this one, we got result. Okay, maybe the employees maybe do some manipulation in such a way that they want to show there is no impurity in that. It's not possible. Whatever you are running, the instrument understood, it will be recorded in 2 ways. One is a dedicated server and the actual server. We can take the printout from the actual server, we can do some manipulation. Whatever you do manipulation, whatever doing, attempting anything, everything will be recorded in that dedicated server. So these regulatory bodies, by sitting in the US FDA, they will access your company, your instrument, what you are doing. Why you are again repeating that experiment? What is the reason? You have to write the reason why I'm repeating that instrument or repeating that same experiment. Whether the chemist or pharmacist attempting doing any manipulation process. So that's why it's a very stringent process. The pharma industry is highly regulated industry, because whatever error something has happened, we can't take it back. It's not possible to take it back. For example, this Crompton fan is manufacturing the fan by Crompton. There is a manufacturing defect. What will happen? So I'm the customer. Okay, I brought the Crompton fan today and installed. After 1 week, there is a problem with the bearing. There is a sound, or there is not running properly. What's happened? I can complain customer care, and they will come and they'll say sorry and replace the fan. And I am also customer also happy. Some inconvenience. Okay, that's everything is fine now. The same thing has happened for any anti-cancer drug or any snake venom, or anything as a BP tablet or any drug molecule which are using emergency for angina during the heart attacks. Can we take it back? So the one of the snake venom is manufactured by some Bharat Biotech. So you are problem with the snake bite. You are giving injection. It does not work out. The patient gone. He died. Okay, the family members go and claim that your the molecule. Is it possible to take it back? Anything? It is completely is a irreversible damage. Remaining all sectors are reversible damage. We can take it back by doing something, rectifying things. Okay, but these kind of medicines, emergency medicines or regular metabolic disorders, we can't take it back. The outcomes is based upon the quality of the drug. That's why the pharma industry is different from the other industries, other manufacturing industries of electronics, electricals, automobiles. These are all different, but pharma industry is different. That's why the pharma industry is highly regulated, not other industries. That's why we have, as a pharmacist, we should be duplicate and there is an analysis in the different levels. The one sample will be evaluated by the 2 to 3 chemists in different places simultaneously. Your result and their results should be matched. They will be evaluated by the manager. You are getting some other result and they are getting some other, something is happening. So that's why it's a highly regulated industry and we need to do work. The result should be very accurate and answerable. Each and every point. Okay, so here just will show what is the basic instrumentation and the part of HPLC. So as you know that I think in interest of time, up to what time? 3 o'clock. What's the time? 2:10. Okay. So as well, I think chromatography. Have you studied the chromatography? Okay. The chromatography now is an accidental discovery actually. Very fast is paper chromatographic technique. There's accidental discovery. Now it is evaluated to LCMS and GCMS, most advanced techniques. Okay. So here in the HPLC, the major parts of any HPLC instrument that contains the first pumps. We have the two parts is the stationary phase and the mobile phase. Okay, mobile phase is a solvent, liquid state here. On the stationary phase, in terms of the column, maybe now we are regularly using the stainless steel columns. Okay, and the principle will be the adsorption. Okay, the separation will based upon the adsorption when you are using the adsorbing agent. Okay, so the principle the HPLC follows different principles based upon the what kind of stationary phase you are using. If the stationary phase is the adsorbing agent, the principle will be the principle of separation is the adsorption. Okay. So this is the basic modes of chromatography: normal phase and the reverse phase. So the normal phase, reverse phase is the chemistry of the molecule nature of the mobile phase and the molecule nature of the stationary phase. Okay. So the normal phase and the stationary phase, most of the times we are using the reverse phase HPLC because we are using the drugs. Most of the drugs are the polar molecules. For the polar molecules, we require the non-polar stationary phase. When it is non-polar stationary phase and polar solvent, it's called as a reverse phase HPLC. 99% of the HPLC methods, what we are using, that's called reverse phase HPLC methods, because we are handling the organic molecules. Most of them are of the polar nature, polar that contains the polar bonds. So these polar bonds are make them to interact the receptor. That's why all the molecules are, most of them are polar that contains the polar functional groups. So these are minimum required to make interaction with other polar group of the receptor. Okay, so that's why reverse flow HPLC that contains the non-polar stationary phase and the polar solvent. Solvent is just like a simple water, and the non-polar stationary phase is C18 column. It's called the reverse phase HPLC. So normal phase HPLC is the reverse. Okay, we are using the non-polar solvent and the polar stationary phase. So based upon the mode, we can classify normal phase and then reverse phase HPLC based upon the principle. Based upon the principles, what kind of the chemical molecule we are using as a stationary phase. For example, we are using the adsorbing agent, the principle with the absorption. We are using the ion exchange material, that ion exchange is the chromatograph is a principle. Okay, we are using the ion size exclusion, means the whatever the column we are using, that is the pore size. So the particles will be separated based upon the size. just like a filtration. Okay, and if it is a affinity chromatography, it is a positive ion mode and negative. There is a positive cations and the negative anions. Okay, so the molecules will be separated based upon the opposite charged molecules will attract. So that is called affinity chromatography and chiral phase chromatography. And we have the different chiral molecules in organic molecules. So these organic molecules, we have the affinity towards the opposite kind of chiral structures. So here, most of the times, almost more than 90% of the studies, it covers first category, adsorption chromatography. Because the absorbing agent we are using, this absorbing agent is sufficient to make separation of the existing samples. So that's why this adsorption will covers most of the times. Okay, so this is a based upon elution. We can take the isochromatic separation, gradient separation. Isochromatic separation means we are using the one solvent system. Entire 4 minutes or 5 minutes or 1 hour, we are using the same solvent system, same ratio. For example, we are using the water as a mobile phase, only water. For entire half an hour, we are using only water. That is called isochromatic, ISO 1. Otherwise, if you mix with the water and methanol 1:1, but entire experiment we are maintaining the same ratio, it is called as isochromatic. Gradient elution means we are using the different solvent system with the different ratios. The ratios with respect to the time. We have started the experiment with only water. After 5 minutes, we are adding methanol. After 5 minutes, again we removed entire water and adding the some benzene solvent system. Means we are using the different solvent systems in the same experiment. So it's called as a gradient elution. So these are isochromatic, and most of the times our molecules maybe better will achieve the better separation in the gradient elution. Because for any HPLC method, the best resolution is we need to complete separation and within the lowest as less as possible, within 4 minutes, within 3 minutes. Because the pharma industry is buying or spending some 30 lakhs, 25 lakhs for instrument, we cannot develop a method for the 1 hour for each molecule. You have to develop better method is always better separation and within the 4 minutes or 5 minutes, as less as possible. Then only the chemist can handle on one instrument daily 50 samples. Otherwise, only you can do 10 samples per day if you sit for the 1 hour per instrument. Okay, so that's why the retention time should be as less as possible, and also we should get a better resolution. Better resolution is better separation of the peaks. Okay. And next one is analytical HPLC and preparatory HPLC. What we are using now, the analytical HPLC. We are using the analytical grade. Preparatory HPLC that contains a large columns for the separation and isolation. So here analytical HPLC, we don't do any kind of isolation. Qualitatively and the quantitatively. What kind the purpose of using instrument qualitatively or quantitatively. So these are the principles depending upon the what kind of absorbing agent you're using. Okay. So this is the basic structure of diagram of the HPLC. The major thing mobile phase. Mobile phase means one reservoir. If it is a gradient elution, more than one reservoir, more than one container, 1, 2, 3 or maximum 4 quarterlet. Okay, and these are pump. The pump because we are using mobile phase. Some energy or some device we should have to pump the mobile phase to the entire system. Column, and this is the column, the stainless steel column. We have now you see this is the solvent delivery pump. It's taking the solvent from here and pumping towards the detector through column stationary phase. So the internal part of the column, internal part of the diameter of the column that contains the absorbing agent, for example. So in the layer, okay. And then detector, maybe is a detector is a PD detector or a fluorescent detector or UV detector. So the detector activity is to analyze the sample by means of the absorbance. Okay, the concentration, if you the molecule that contains the most concentration will get the high absorbance, otherwise the low absorbance. Okay, so now we are injecting here somewhere here. Okay, so the molecules will be make it available to the column. The column length is up to 30 centimeters. Short columns, this one. Gas chromatography columns is in the meters. Okay, HPLC columns are the maybe the centimeters. So this is the column inlet and column outlet. So for example, we are injecting the sample that contains the 4 components A, B and C. all A, B, and C will interact the adsorbent here. A elute first, and then C elute after 1 minute, C elute after 2 minutes, and B elute after 5 minutes, and finally D compound D elute at the 10 minutes. So the resolution each after 1 minute we'll get one peak, after 2 minutes we'll get the second peak, third peak, fourth. All the 4 components are eluting from the column and 4 peaks are separated. Okay. So this is a simple one of the typical image of the water. Waters means is a brand name. HPLC, one of the leader in the world, is a Waters is a company name. Waters is a manufacturers of some of the equipment, includes they are leaders in the HPLC. This is the pump 1. This is the pump 2. This is a dual pump HPLC model. Okay, and these are the reservoirs. Okay, now from the reservoirs to the pump to the air injector will be there, and then make it available for the detector. You have different detectors. If you want to use the UV detector, you can connect to the UV. The outlet of the column you can connect to the UV, or if you want to use the other detector, you can detach it and you can connect to the detector. Depending upon the type of sample, you can utilize different detectors based upon the application. Okay, so these are the different principles of the separation, how it happens. Ion exchange, size exclusion, adsorption, different phases, normal phase and reverse phase, and isochaotic and gradient elution. So I will show some. So these are the type of surface coating in the HPLC, reverse phase HPLC column C8, C18. What is the meaning of C8? C8 that contains 18 carbons. It is in the non-polar nature. That component what you are using as adsorbent contains 18 carbons, 8 carbons. Okay, so these are the two we are using the non-polar nature, and these are the things, and silane and amino groups is polar nature that contains the polar functional groups. So these are type of materials are as a binding agents in the stationary phases. In the reverse phase, these are the materials in the normal phase and the reverse phase respectively. Okay. And these are the materials we are using as a stationary phase. Okay, so here the ideal characteristics is always we are using the mobile phase is the HPLC grade solvents. For entire HPLC experiment, even for the sample preparation also, you need to use the HPLC grade solvents. And mobile phase also HPLC grade solvent. This is a purity. HPLC means maybe sometimes 10 times more than the regular solvent. For example, methanol is 300 rupees, HPLC grade methanol maybe 3000 rupees per liter. Okay, so that is the quality or the purity of the solvents, because we are sending and all these mobile phases we are running through the column. The column there's a very sensitive stationary phases. If the solvent contains impurities, they will lock. So the column will spoil. If you accidentally, if you run with some other laboratory grade solvents also, that affects the column. Some of the constants we can wash from the column, some other they will bind irreversibly, permanently. So that's why we always use the mobile phase system, pure mobile phase, that is a HPLC grade mobile phase system. So these are the characteristics: purity, detector compatibility, solubility, low viscosity, chemical inertness, reasonable price. Price also important. Okay, whatever the method we have developed, there is a better we should develop method with the low cost solvent. Okay, and low corrosive solvent. Environment also important. And low viscosity. Why is it like viscosity? If the solvent is high viscous, it is difficult to pump. Actually, is running. The solvent is running. Is a flow also important? If it is highly viscous, then is a problem with the elution process. It will take time. Okay, there's a column packing materials, and these are the some of the flow ranges and typical flow rates. And the cost of the column is typically between the $1,000, maybe the 60,000 per column. Okay, and we can use these columns repeatedly for the time based upon that. You're using how carefully you're using, we can use these columns for the, we'll get number of experiments for the same column. Okay, so I will stop with this presentation with this. So this is actually happens in the column. If you look at this one, this is a column. Okay, and we utilize the mobile phase is M. Assume that mobile phase is M, and stationary phase is S. We are using stationary phase. Yes, maybe some adsorbent silica gel. Okay, mobile phase M, it is the water. We are using mobile phase is the water, and stationary phase is silica gel. Yes. Okay. Now this is the components we are running. This is maybe the C is a paracetamol, and A is some of the constituent of the paracetamol as impurity. Paracetamol, we are estranolide, we are just synthesizing from the some estranolide derivative. Some part of the estranolide is not converted into the final product. We got some impurities. Okay. So our sample contains C and A. We are injected C and A. So these C and A interact with the mobile phase as well as the stationary phase. So you see the compound C, we have an attractive force towards the mobile phase, and so both it has an attractive force towards the mobile phase and stationary phase. At the same time, A also it has an attractive forces towards the mobile phase and towards the stationary phase. But there is a net attractive force, means whether the C contains a more attractive towards the mobile phase or more attractive towards the station. That will decide how much time this C is going to spend in the column of 1 centimeter or 10 centimeters length. For example, the C is having a more attractive towards the mobile phase. They will travel very fast with the mobile phase. Maybe the distance is this much, column distance. Maybe it's 20 centimeters. So the C has to travel 20 centimeters. If the C is having a more attractive forces towards the mobile phase, because the mobile phase is running. So the C also will travel comfortably along with the mobile phase and will elute from the column first. And then it is make it available to the detector, and we will get the peak which is responsible for the C. We will get the C peak. Then A is a more responsive, more attractive forces towards the stationary phase, not mobile phase. Then A will travel slowly because stationary phase is stationary in the column. So A is attracting more attractive force towards the stationary phase. It will travel very slowly. After 10 minutes or after 5 minutes, it will elute, and we will get the peak A and B. So ultimately, we will get the these are the columns, the spectra I'm looking at. Yeah, you get the spectra like this. Okay, so maybe eluting after 5 minutes, retention time is 5. Okay, B is eluting after 10 minutes, retention time is a 3. And the area under the curve is a concentration. If you use the sample contains a more quantity of A, you will get the high peak. Area under the curve is a quantity, and their qualitatively is retention time. The time is required to elute the compound from the column. It's called as the retention time. Okay, the peak, the area under the curve is directly proportional to the concentration, the quantity of the component that contains in the given sample. Okay, so here these are the two components eluted, and we'll get the spectra. Then the interpretation of the spectra will get the assay of the sample. The assay of the sample is like if you take any dosage form as per IP, it should not be more than 99% to the 101%. Within the limits or not. You have to check within limits, then it is a path, then it is a as per the regulatory bodies, what is the assay within the limits. Okay, we have to use their prescribed methods only by prescribed procedures. Okay, so very first, what is the important thing is like the method development. Method development means whenever anyone doing the first time, you need to develop a method. Once the method is developed, the regularly it's the same method. You're usually in the QC and department or QA department, they are following the regularly the same method. Okay, already it is an existing method, and as per the regulatory board, this is the method you have to follow. So the stationary phase column is fixed, mobile phase is fixed, all the parameters are fixed. They're using the same method, same chemist, same preparation of the solvents, the entire like the same methods and same documentation. If you are doing any new kind of method development, then you have to check with, you have to try with the different mobile phases. For example, the method contains 1:1 ratio of water and methanol. How do you know 1:1 ratio? Because already it's done, already published, already somebody is developed that method. They have reported 1:1 ratio of This one, when you are doing the very first time, you have to trial. You have to trial with based upon the chemistry of the molecule. You have to look at the chemistry of the molecule, whether it's how many polar functional groups, how many non-polar functional groups, what kind of solvent is suitable, what kind of column is suitable for this, what kind of detector suitable for this. We need to all these things work out thoroughly, and we have to develop a new method, and all these methods you have to validate it after that. Then it is a method is developed and validated for all the parameters. We have different validation parameters. If the validation is completed, then we can publish that method. So anyone will utilize or any company or anything, any party can utilize that method for the determination of the assay or perform the assay of the particular drug. Okay. So you have any questions? So your very important thing is like handling the instrument, not only the HPLC, any kind of analytical instrument, any kind of analytical or any kind of the sophisticated instrument in any department. The very important thing is you should be able to interpret the final result and spectra, and you understand the results. Anyone handle the instrument. If you train one day, you can anyone able to handle the instrument. Only just you are giving the dose, wait for some time, 15 minutes, you'll get some result and take print and you will get the paper. The thing is here, the understanding what is the selection process of solvents, mobile phase, selection process of stationary phase, selection of the pressure, pump pressure. After that, they will get the result, understanding the results, interpreting the results. These are all important. These are all the unique features. We need to acquire the knowledge as a chemist or as a pharmacist. It's very important. Your growth is depending upon your understanding the concepts. You should work intellectually rather than the physically. So 95% of the people will work physically, will submit the sample and they will get. So that's called that will decide your growth. Each and every method, when you look at the method, you should understand the concept. Do you have any questions? Any questions? No, everyone is sleeping? Then బయట వాళ్ళని ఎలాగ అడగరు? Yeah, you can ask anything, yeah. Anyone? You not get any curiosity? Retention? Retention volume means the volume, there is a two types of volume, wide volume and the retention volume. The wide volume means there is a space when you're injecting the time, the very first time, okay, so you will get some gap. It's called as a wide volume. Okay. The retention volume is the volume of the sample, okay, at the retention time. Okay. So this is called as a retention volume and the wide volume and the retention time. So these are all these parameters when you are calculating the resolution of any particular peak. Yes. Calibration. So we have the different methods of calibration of HPLCs. So each calibration, for example, as per you see the IP, okay, Indian form, we have some pharmacokinetic techniques. Any instrument, not only HPLC, there is a calibration methods. Okay, so means even the balance also. You're using the balance. You're using the pH meters. Okay, and you're using the some of the like nephelometry and turbidometry. So there is a specific methods of the calibration methods. You need to repeat that calibration. But we are not as a student in the laboratory thing. We are not regularly doing the calibration. But in industry, every time we need to calibrate by specific method, and we have to write the report. So this method is this instrument is calibrated by means of so and so method, and the so and so chemist done the calibration method, and this so and so. So this one, each one we can't do the same method calibration for the reverse phase HPLC and the normal phase HPLC, or kind of the materials you're using, kind of the adsorbents are using. Okay, so we have the specific methods of the calibration. In layer TLC is like, yeah, thin layer chromatography is a manual technique. We have HPLC also, but thin layer chromatography is just like we have the plates. Okay, then we have to give a sample on the plate, then we'll develop, we'll get the two components are separating. We are not just identifying, we are just looking at the samples under the light, whether they're separated or not. Okay, based upon the RF value, what are those components? Okay, but here this high performance HPLC, we are not only separating, we are also identifying. Qualitatively identifying, this is the quantity that contains, this is the retention time, this is the based upon. So the retention time also, we have to fix the lambda max of that UV detector. For example, you are evaluating the caffeine. Caffeine lambda max is 72. The entire experiments we are doing at the 72. Okay, we'll get the peak for the caffeine at the lambda max of the caffeine. So the TLC is, for example, you're adding some amino acids sample. Okay, you're taking the amino acid that contains the glycine and the alanine both. Okay, that is a mixture. The your goal is to identify the what kind of amino acid present in the given sample. You are adding the mixture and you are developing the sample, you're spraying some inherent reagent. These two peaks are visualizing and calculating the RF value. Distance solved by the solute and distance solvent. Usually, the particular method, when you're using the methanol as a solvent, silica gel G coated 1:1.5 mm thickness silica gel G, and the what we got the 1.2 RF values maybe is the glycine. Okay, and this one alanine get the 1.4 RF value. Okay, so it's based upon the TLC is a very nominal, a simple technique. Okay, and HPLC is a completely instrumental technique and is a sophisticated technique. Is run by the software. Suppose carryover effect. Carryover effect. For example, carryover effect and no calibration. What's carryover effect? I don't think. We see you're telling the carryover effect, how it is affecting on. That's the procedure. Okay, the procedure of the okay. You are telling the you are talking about the procedure of some HPLC technique. There is a calibration. The calibrations you have the different routes, right? So one of the technique that can say carryover effect is, for example, the affinity chromatography. That's what. For example, HPLC. We cannot consider all the HPLC in the same category. Okay, we have some 5 to 6 categories of the principles. The affinity chromatography is a different principle. Okay, and the adsorption chromatography is a different principles. Okay, and maybe is like chiral is a different principle. The affinity is a different principle. The affinity is only also opposite to the filter paper. Means the pore size that contains small particles. The smart particles will lock into the pores and will travel slow. The larger particles we can never lock into the pore size. Will travel fast. Is opposite to the even the chemical method of separation like the chiral or adsorption. Okay. So what you are asking, there's some carryover effect. Okay. That's what calibration. The maybe is a calibration of the adsorption chromatography HPLC. That's what calibration of absorption chromatography. The calibration, because why I'm telling the absorption chromatography equipment, most of the 90% of the HPLCs will follow the columns of the C18 columns. in the reverse HPLC columns. So that's why there is a generally that contains most of the percentage. Okay, that how you have encountered the carryover effect. That is what's my expecting. Okay. Yeah. Anything related to detectors, anything there's a solvent systems. Yes, proteins. Yeah, we have so many like the chiral detect like for especially proteins. The major difference between the small molecules and the proteins are the larger in size, biologics. Okay, so that contains these absorption will not work out. These kind of detectors will, these kind of columns will not work out. So that is affinity chromatography is one of the and mass is the one of the detector. Mass spectrum is one of the detector. Spectrometry is the detector is a mass now. So, for example, small molecules we can handle with the UV or PD detector. For the proteins, for large molecules, because that contains a mass spectrometry, that contains a positive ion mode and negative ion mode. Even you can go with 15,000, 16,000 molecular weights also. And also the proteins, the major problem is the energy. They are very sensitive molecules. These are all organic molecules are very stable molecules. The proteins are very less stable molecules. So yeah, the mechanism is similar like this, but the column, what kind of column? The column, only the difference is column. We can use the same machine what we have with the protein separation also, or the protein analysis also, but we require the different column. We have the C18 and C18 columns. So those experiments which are related to the C18 and C8, we can do perform. If you want to the protein kind of what kind of protein it is, what kind of things, then you have to select the suitable things. So how can what we do? 2020. How many are there? Take it to- okay, so- Take it.